Hello everybody and welcome back to Ed Puzzle Notes. Today we're going to be going over protein synthesis or how proteins are made. Now remember, DNA has the code for making proteins. Remember it was stuck in the nucleus, it can't get out, so it has to hand off that code to messenger RNA, which then takes that to the organelle called ribosomes, which then can make proteins. Remember, the little tRNAs, those don't carry the code from DNA, but those guys carry the ingredients or the building blocks of proteins called amino acids. All right, there are two steps to protein synthesis. Step one is called transcription. And basically, in a tiny nutshell, transcription is the process of doing this. It's the process of making a messenger RNA from DNA. The second step is called translation, and that is where proteins are made at the ribosome with the help of messenger RNA and tRNAs. But we'll get more into that here in a second. Alright, so once again, here is messenger RNA or mRNA. Now for the test, you're not going to need to know everything about mRNA, but you need to know at least three things. Number one, what does it look like? Well, it looks like that. You will have to recognize it on a test, from a picture, to say, hey, that's an mRNA. Well, how can you tell? Well, you can see that it is single-stranded, so it doesn't have two strands like DNA. DNA would have some more bases attached to these guys, with sugar and a phosphate, so it'd be double-stranded. That's single-stranded, so you know it's RNA. You also can see it has uracil, or U, so you know it's RNA, mRNA. And remember, DNA and RNA, they talk in three-letter words. So each little three bases or three letters, remember, in mRNA that makes a codon. Now, how is it made? Remember, it's made in the nucleus because it copies the code in DNA. And that process of copying the code in DNA in the nucleus, that is called transcription. Transcription. And finally, what does it do? Well, remember, it copies DNA's code. DNA is stuck in the nucleus, so mRNA is able to take that code to the ribosome to make proteins. All right, now this will be a tRNA over here. Once again, you're not going to have to know everything about this thing, but number one, what does it look like? It looks like that. It's got kind of a weird shape, looks kind of like a candlestick holder. mRNA looked like this, a single-stranded with bases like that. So that's what mRNA looked like. tRNA obviously looks way different. It kind of looks like a T if you did that. Also know, here's that three-letter word that RNA loves to talk in. This one says GAG, guanine, adenine, guanine. And remember, this is not a codon on a tRNA. It's called an anticodon. So remember, mRNA, codon, tRNAs, anticodon. And what does it do? Well, this is important. It carries what's called amino acids. Remember, that's the building block of proteins, amino acids, and it takes those to the ribosome, where proteins are made. So this particular tRNA, you can see it up here, it's carrying the amino acid leucine. LEU is for leucine. Now when it gets to the ribosome, this anticodon down here, right down there, the GAG, guanine, adenine, and guanine, it's going to match up So that's the anticodon. It's going to match up with a codon on an mRNA molecule, and then it's going to leave its amino acid behind. So, what would the codon be that would match up with guanine, adenine, guanine? Well, it's always the base pairing rules. G goes with C, A goes with U, because it's RNA, and G goes with C. Alright, so this slide should be all review. Remember, DNA carries our genetic code, our genetic information, so it gives us all of our traits. And those traits are because of different proteins that are made at the ribosome. So the different proteins is what gives you all the traits you have. Your brown hair, whatever color skin you have, eye color, whatever. That all has to do with whatever proteins are being made. And the code for making these proteins are found within DNA. Also remember that it is a bunch of amino acids that are linked together to make these proteins. Some more review. Remember, the reason why we need messenger RNA, mRNA, is because DNA is stuck in the nucleus. DNA is stuck in the nucleus, so it has to hand that code off to messenger or mRNA. And it is that messenger RNA that actually takes the code from DNA to the ribosome to make the protein. Alright, so transcription. You're not going to need to know the steps like DNA replication. Here's what I want you to know. Transcription right there. Just write this. It is the process of making a messenger RNA or mRNA from DNA. That's all you need to know. Now, if you want to see how it occurs, this is how it happens. DNA in the nucleus unzips, and then with the help of enzymes, these RNA bases are attached according to the base pairing rules. So A would go with T, C would go with G, U would go with A would go with T, and so on. So it keeps going until it reaches the end, and then this mRNA molecule, this green thing right here, will travel to the ribosome to make a protein. Alright, so once again, transcription is just the process of making mRNA from DNA molecules. So there's a tiny, this is obviously DNA. It's been unzipped because it's single-stranded right here. There would be more bases over here, but they've been unzipped. You can also see it's deoxyribose sugar. So if we're going to make a messenger RNA, it's just the basic base pairing rules. G will go with C. C will go with G. A will go with not T, but U. T will go with A. And there's a little tiny mRNA molecule. Now these ribose sugars and the phosphates, those will also be bonded onto those guys. And this guy will leave to the ribosome to make a protein. And this slide's pretty much worthless. But once the mRNA is made from the DNA, it's not going to stay there. It's obviously going to leave the nucleus. It's going to leave the nucleus and heads to the ribosomes. So there it can make the protein. And the DNA strands, obviously, they're going to rejoin back together. But you won't need to know that. Just know that the mRNA will leave the nucleus and head to the ribosome to make a protein. Now the second part, translation. This is the part where we're actually going to build a protein from the code in the mRNA molecule. Now obviously, first thing we need to happen is the mRNA needs to arrive at the ribosome, because we won't know how to make the protein until that mRNA arrives. Also, remember, the ribosome is made up of the other type of RNA called rRNA. That was, the one I said was least important, but try to remember that one. The ribosomes are made of rRNA. Now, proteins will begin translation or begin being made on what's called the codon for start. So remember, a codon is 3 bases or 3 letters on an mRNA molecule. 3 bases in a row in an mRNA. And the start codon is always AUG. Adenine, uracil, guanine. How is that easy to remember? Well, when does school start? It starts in August. Boom. So proteins start at AUG, school starts at AUG-UST, so that should be easy to remember. So now mRNA has arrived at the ribosome, so it has the code, it has the instructions for making the protein. Now all we need are the ingredients to make the protein, the little building blocks, and those are amino acids. We need those things to come to the ribosome because we cannot make proteins without amino acids. Well, remember, it is tRNA's job to bring those amino acids to the ribosome. The tRNA's 3 letters or 3 bases, which we remember are called an anticodon, is going to match up according to the base pairing rules with a codon on mRNA. It will then leave that amino acid behind and it will be added to the protein string. So protein, all it is a bunch of amino acids bonded together. Now, how many bases make up codons and anticodons? That's easy. It's 3. Remember, 3 bases in a row on an mRNA makes a codon, and 3 bases or letters in a row on a tRNA makes an anticodon. How do the bases match up? The same stuff we've been doing for the last week, according to the base pairing rules. So let's use that start codon of AUG. Remember, this is going to be on mRNA. So what would the tRNA anticodon be that matches up with AUG? Well, A doesn't go with T because it's RNA, it's going to go with U. U would go with A, G goes with C. Continuing on with translation, we've pretty much done. This is the process of building a protein from the code from mRNA, which it got from DNA, with the help of tRNAs, which bring amino acids to the ribosome. Remember, ribosome, that's where proteins are made. Now, these amino acids are going to be linked together not by hydrogen bonds, like which holds together DNA, but what's called peptide bonds. Now that'll be like one question on your test, so don't kill yourself trying to memorize that, but it's peptide bonds. And that's why they call, another word for proteins, is they call them polypeptides. Polypeptides, so you might see that word sometimes, and that just equals a protein. Now, these tRNAs will continue to drop off amino acids. They'll keep dropping off these amino acids, making the protein longer and longer and longer and longer and longer and longer and longer. It'll keep going until, remember we had AUG was the start codon? Well, there's also several different codons that tell the ribosome stop. And boop, it'll cut the protein off right there, and that protein will go off into the cytoplasm to do whatever job it needs to do. Now this is my favorite diagram when it comes to translation. So this is translation. We are making a protein, and this is it. This is where it's called a peptide chain. Here's the protein growing longer and longer right here. Here's a tRNA right there that used to have an amino acid, but it already dropped it off. It dropped off the amino acid tryptophan, TRP right there. Now you can see its anticodon here of ACC, it matched up with the codon of UGG. That's how the ribosome, where proteins are made, knew to drop off tryptophan. Now this anticodon on this guy is UUU. Well, the reason it's there is because it matches up with the codon of AAA. And it's dropping off the amino acid called lysine. This next guy that's putting on his amino acid right now has the anticodon of CUA, and that matches up with the codon of GAU. And it's leaving behind the amino acid asparagine. Now the next guy that's coming in, he's waiting his turn. So this is, the code right here. Here's the next codon, UUC. Well, U goes with A, U goes with A, C goes with G. Oh, there it is right there, AAG. It's going to match off with that codon, and that guy's going to leave the amino acid phenylalanine. So it is the order of these bases, the order of these codons, which determines the order of these amino acids that are being added to the protein chain. So if you change these codons, if you change that UGG and made it like GCU or something, you would not get tryptophan. You'd get a different amino acid. So these codons determine the order of amino acids to be added to the protein. How to build the protein? Whoa, I don't know if I've said this before, but proteins are made from the monomers or the building blocks called amino acids. So you stick a bunch of amino acids together. Stick, stick, stick. Remember, those are peptide bonds that are bonding those guys together. So a bunch of amino acids bonded together with peptide bonds, you get a protein. Boop. There you go. And if you want to determine the amino acid sequence in your protein, like what order, what order are these amino acids? Well, look at the mRNA codons, and they will tell you exactly which amino acids are in which order. To do that, we're going to use what's called a codon chart. Now, we're going to do this again on the next class period, so you don't really need to take notes on this right now, but you're going to have to learn how to use these pretty quick. All right, so like I said, we're going to go over this again the next class period, but it's never too early to start learning this thing. So this is what's called a codon chart. Codon. Remember, codons are on the mRNA molecule. So this is not an anti-codon chart. This is a codon chart. So we're looking at the three letters or three bases on an mRNA. So let's start with the start codon, AUG. Which amino acid would show up with the codon with the bases AUG? Well, to use this codon chart, it's pretty easy. First, you look at the first base in AUG. The first base is A. That's the first one. So then you look over here. First base, it's not a G, it's not a C, it's not... Oh, the first base is an A. All right, sweet. So now we know the amino acid is somewhere in this column right there. Now the second base is a U. So now we're going to look up here on top, the second base. All right, it's not a C, it's not an A, the second base, not a G. Oh, it's a U. So now we know it's going to be somewhere in this box. Now to figure out the specific amino acid, you look at the third base, G. All right, third base over here. Now we know it's in this row because of the first base. So, G, it's not an A, it's not a C, it's not a U, it's a G. So then we just look across, and voila, there it is. Methionine is going to be our first amino acid for the start codon of AUG. Let's try one more. Let's just make up one. Let's go UUC. All right, the first base is a U. So we go over here, first base is a U up here. So we know it's somewhere in this long rectangle right there. It's in one of those. We look at the second base. It's also a U. All right. So now here's the U right there. Now we know it is somewhere in this box. And to find out what exactly it is, we look at the third base, which is a C. So then we look over here on this side, the third base. It's not a G, it's not an A, it's not a U. Oh, it's a C. Then we just go right across, PHE equals phenylalanine. So, I don't know, you're not going to be experts on using a codon chart right now, but we're going to practice it, but hopefully that helped you out a little bit. All right, thank you very much for listening, and I'll talk to you next time.